thank you for being willing to listen to me talk about one of my favorite topics for an hour. One can talk about this for days and days and days, but I will limit myself to an hour, and there are some things that I will not talk about, and I made a list, otherwise we would be here forever. First of all, we know what DNA is. It's an acronym for deoxyribonucleic acid, which is the instruction molecule in cells. Okay. It's named for partly its components. Deoxyribose is part of it, which is a sugar, and nucleic acid tells you where it is. Most of it is in the nucleus of cells, and it has the chemical property of being an acid as opposed to a base or a neutral substance. So that's where they came up with the name, but for our purposes, we'll just call it DNA. Okay, here's what we're not going to talk about. And maybe this can be the subject for another talk if you don't get tired of listening to me. Okay, we're not going to talk about Ancestry.com or 23andMe, because that's a whole nother story. We're not going to talk about CSI and DNA fingerprinting. That's another story. And we're not going to talk specifically about genetic diseases. That's another story. Okay. What I am going to talk about is the location of DNA and its structure and its functions. And in particular, I'm going to try to give you a foundation so that you can understand why the four women I'm going to talk about made contributions to our knowledge and understanding about DNA that are so valuable and so important. Now, there are thousands of people that have worked on DNA and helped us move forward with our knowledge, but these are four that I picked out that have made what I consider to be big steps forward, and I wanted to share them with you. You see my quote up here, that there's no problem in science that can be solved by a man that cannot also be solved by a woman. So, science is a passion, a mindset, a way of thinking, and anybody can have these things. And sometimes you are born with that interest, and sometimes you acquire it as you move on through life and things catch your attention. But if you are a puzzling kind of person who likes puzzles, then science is probably something that appeals to you. We're going to start with some history. Anybody recognize Gregor Mendel? Okay. All right. If you've ever talked about or heard about genetics, he's probably the first person you heard about. And he was working back in the 1850s as a gardener in a monastery. He was a monk, and they gave him the assignment of raising the food for the members of the monastery to eat. And in addition to doing that, he also was interested in plant breeding. And so he did some things that others had done throughout history of picking parents that had a particular characteristic that they were interested in and breeding them with each other in hopes that the offspring would have it. And for hundreds of years, people had been noticing that there was a relationship between the parents and the offspring. Could be height, could be number of kernels of corn on an ear of corn, could be, lots of things that you might select for. But what he did was pay attention and do something intentionally. So he was working with garden peas, and they are usually self-pollinating. Their flowers are such that the petals hide the male and female part of the plant so that the pollen falls on the female part and you get offspring that are pure or that are having the same information from both parents. Okay. What he did was to cross-pollinate, to peel away the petals from one flower and take the pollen with a paintbrush and paint it on the female part, the pistil, of a different flower with a different characteristic, and then look to see what the offspring looked like. So he crossed a white flower plant with a pink flower plant, and all of the offspring were pink. And so, oh, well, that must be that only that one parent is contributing to this offspring. He did the next step. He allowed those offspring to self-pollinate and discovered that 75% of the offspring were pink, but 25% were still white. And so that showed that definitively for the first time that both parents contribute to an offspring, and that information is not lost or blended, it remains discrete, so it can be handed on separately to the next generation. Well, he did this with 7 different characteristics of peas, just to make sure that it wasn't an anomaly when he was looking at flowers. And he did more than 3,000 pea plants, counting and checking all of this. So this was a big step forward, and demonstrated that each parent contributes genetic information to the offspring. You would think that this would be big news in the science community, but his work went undiscovered for about 30 or 40 years. So we're coming to the end of the 1800s. But by then, microscopes had gotten good enough to be able to watch cells divide, so cells produce offspring, and to look and see something was separating. So something was following the pattern of being contributed to both offspring, or from both parents to an offspring. Scientists named these things chromosomes because they absorb a colored dye, chromo, and somes means body. So chromosomes, these were colored bodies when they looked at them under the microscope. Well, chemical analysis proceeded then on into the early 1900s to determine that there were 2 different kinds of molecules making up the chromosomes. Now, do we know what molecules are? Okay. We know that the smallest building block of things are atoms, okay? And molecules are groups of atoms connected or bonded together. Water is a molecule. You know its formula. H2O, so 2 hydrogens and 1 oxygen. There's 3 atoms in a water molecule. So for living things, found in living things, organic molecules, there are 4 categories of them. And you've heard of them because they're on the side of your food packages. There's carbohydrates and proteins and lipids, which is a fancy name for fats. And then the one you probably have not seen is nucleic acids, because we don't get much nutrition from that. And that's what's on the side of the food packages, is the nutrition things. So what was discovered as scientists were taking apart these chromosomes is that there were proteins and nucleic acids there. Okay, well, where's the genetic information? Is it in the protein or in the nucleic acid part? And this is what scientists were working on well into the 1940s. These 2 molecules are similar to each other in their structure. They are polymers made up of monomers hooked together. Mono means one, poly means many. Okay, so hooked together like the train that I've put up here. Linear molecules, okay? The monomers in proteins are called amino acids, and you've probably heard of that before. Sometimes we're recommended that we take a amino acid supplement, and so you might have gone into CVS and seen little bottles that say this amino acid or that amino acid. And there are 20 different ones of them. Since you're not biology students, you will not need to know their names. Nucleic acids, on the other hand, have only 4 monomers, and you probably have seen those letters before. A, C, G, and T. Those are initials for 4 different molecules that are part of these monomers. So 4 different monomers, each one has its own difference to it. Okay. So there are 4 of these monomers linked together to make nucleic acids, but 20 different ones linked together to make proteins. And think of them as being like letters in a word, that they can be in all different combinations and all different lengths. Some words are 3 letters long, some words are 20 letters long, and same way with the nucleic acids and the proteins. They're not all the same length and they're not all the same order of monomers, but they're all have these same monomers. Okay, so where is it? Come to find out, it was in the DNA molecule. Some great experiments happened in the late 1940s. The missiles install experiment, for example, which I will not tell you details about, but someone may have heard of this before, definitively showed that it's the nucleic acid that has the instructions, not the proteins. Proteins, we now know, are regulator molecules and kind of a support structure for the DNA. Okay, so where is it? This is what I'm going to talk about. Where is it? What's its structure? And what's its function? Most of it is in the nucleus of a cell. Do you know what that is? Okay. When you look at a cell, there's a big structure inside of it, the biggest of the structures inside of it, and that's a package, kind of a bubble in which all the DNA is located. Now, let's talk about chromosomes. How many chromosomes are there in human cells? Anybody know this? Okay. Would 23 be a hint? 23 times 2. 23 in a set of different ones, but 2 sets in each cell, one that came from mom and one that came from dad. Okay. Why did I say that? That's in the nucleus of the cell. There is some DNA that has been discovered in other little structures outside of the nucleus. If you've ever heard of the mitochondria, that's where there is maybe 2% of the amount of DNA that's in a regular cell, just a little bit. That's another whole story for another day to tell about, and that was a discovery by a woman also, Lynn Margulis, but I'm not talking about Lynn today. Okay, so that's where it is. Now we're going to talk about its structure and its function for the rest of our conversation here. So it's in the nucleus, and it's in individual packets called chromosomes, and if you take the chromosome apart and take the protein away from it, that's what the DNA molecule looks like, this twisted ladder kind of thing. But they didn't know that in 1950. The 4 women I'm going to talk about today are related to 4 different topics that are part of DNA. We'll talk first about Rosalind Franklin, who was important in determining the structure of DNA. Then we'll talk about Elizabeth Blackburn, who was important in the replication or the copying of DNA. And then we'll talk about Jennifer Doudna, who is leading the way in gene editing, which means taking out a damaged part and putting a new part back in. And then finally, protein production. Kathleen Carrico is who we have to thank for the mRNA vaccines that were part of our COVID protection. Okay, so the race is on in 1953 to find the structure of DNA, because whoever finds it's going to be famous forever. And if I were to ask you who you would associate with the structure of DNA, what would you say? Watson and Crick. Okay, Mr. Watson and Mr. Crick. Okay, There were 2 questions about this on Jeopardy the other night, so this is still very much in the talking. So the race was on. Watson and Crick were at Oxford in England, and Crick was the lead scientist, and Watson was a newly graduated PhD from Indiana University that came to work in Crick's lab. And they were building models. The Tinker Toys. Did you play with Tinker Toys when you were a kid? Okay. Their model looked like this, with the support structure in the middle and the A's and T's and G's and C's sticking off to the side, kind of like a bottle brush. That was the best picture of a bottle brush I could find online. So that's what they were doing. Over at Cambridge, Wilkins was the major scientist, and Rosalind Franklin was also in the lab, but he was the senior scientist there. She was going at it from a whole different angle. She was crystallizing the DNA. So she took the DNA and then crystallized it and then took X-ray pictures of it, shot X-rays through it. And this is the picture that finally made the decision. So it shows the dark things on the inside. Those are the A's, T's, G's, and C's instead of on the outside. And the heavy dark areas are the support structure. So Watson and Crick had it inside out. So like this, but later research showed that one of them is upside down compared to the other. It's what's called anti-parallel. And this becomes important later when we talk about how the copying occurs to make new DNA. So she had this picture, along with others, in a file folder on her desk at Cambridge when Watson came over nosing around to see what they were doing, because they weren't having any luck with their Tinker Toys. And when she left her office, he went right over, opened the file folder, took one look at that, and immediately recognized what he was looking at. Because of what he'd been building, he saw, oops, I got it inside out. He rushed back to Oxford, and they published the paper in less than a month, and as of this Friday, it will have been 73 years since Watson and Crick got the credit for this, and it was years before Franklin was even mentioned. I mean, if you look at textbooks, even in the early 70s, she's not even there. But things are getting better, and we're talking about her more and more, and here she is. She was British, and her X-ray diffraction made all the difference. Now, this is the Tinker Toy model that Watson and Crick were building, and it was 3 stories high. So think about, you know, if we had 2 more Longwood rooms on top of each other, they had built a model all the way up, but inside out. And then when he got back with that picture, it was like, oops, okay, flip it. And there it is. This is Watson over here. Watson died in November this past year at age 97. Wow. He was not a pleasant man his entire life. There are lots and lots of stories about the additional research that he did and people that he interacted with, and not someone that you would have enjoyed having around. But he had good realization of what he was looking at, because she had been looking at that too, that picture, and it hadn't clicked with her either. So in 1962, the Nobel Prize went to Watson and Crick and Wilkins, but not to Rosalind Franklin, because it's not given posthumously. And she had died in the late 1950s of ovarian cancer, probably as a result of the X-ray exposure that she had in doing the research that she did. So we remember her fondly and often, every chance I get. So that's the structure of DNA. Now let's talk a little bit about its function. It's an information, not molecule. That's its whole purpose, information. And it can do two different things. It can make a copy of itself, replication, and it can be a pattern for making proteins, which is what is really, really important to all of us. This is a little picture of the life cycle of a cell, and the point I want to make by putting it in there is that the DNA is used as a pattern in two different parts of the life cycle. So in the G1 and G2, it's making proteins, and in S, it's replicating. So the pattern's being used for two different things at different times in a cell's life cycle. Make sense? We're good? Okay. This picture also lets me talk a little bit about protein production, and I'll talk more about it in a few minutes. Okay. The DNA molecule is double-stranded, 2 strands. One side is information, one side is placeholder. Okay. And so when a protein is to be made, it opens up, the 2 sides separate from each other, unzipping, because it looks like a zipper, and one side is used as a pattern to make another kind of nucleic acid called RNA. You heard of that before? They are similar in that the sugar is very similar. It's ribose instead of deoxyribose. Deoxy would mean 1 less oxygen in there. Okay. And for the bases or the letters, instead of T, RNA has U. A little bit different structure to it. So they're similar, but they're not the same nucleic acids. So one side of the DNA is used as a pattern to make RNA, and then the RNA is used to assemble the monomers of the protein into a protein polymer. So that's how protein production takes place. This is a real simple version of this. Again, we could spend a whole day or 2 talking just about protein production. But I wanted to show you that as an introduction, because when we talk about Kathleen Carrico, that's really important. So, what happened? Okay, so we talked about Rosalind Franklin and why she was important in the structure, determining the structure of DNA. Now we're going to talk about Elizabeth Blackburn and her contribution to what we know about replication. So we're on this side of that picture talking about making a copy of the DNA. And why do we need to do that? Because when the cell divides, each cell should get a complete set of what was in there to begin with. So when your cells divide, 46 chromosomes go into one cell and the new cell. So you got to copy it every time you're going to make a new cell. And we make new cells all the time, all the time. All right, so now I want to talk about Elizabeth Blackburn. Okay. She discovered that at the end of chromosomes, there are regions that are repeated over and over again. The sequence of nucleotides, or the bases, just lots of the same sequence over and over and over again. This is protective for the cell because when replication occurs, because of the anti-parallel structure, this end and off of the other end that doesn't get copied. And that's shown in the picture up here, that the new strand is red, and then there's this little yellow piece at the end that is not copied, and so when they're through, you end up with chromosomes that are a little bit shorter than the parent chromosome. The fact that there are repeated sequences at the end isn't a problem for the cell, because that little piece that's missing, there's already more copies of it on the new cell or on the new chromosome. So you can chop off the end many times and still not lose important information. So it's kind of like the end of your shoelace. There's a protector on the end, and if you snip off a little bit of it, there's still plenty there to hold your shoelace together, and then a little bit more. But eventually, you do get to important information, and when it's left off of replication, then that's a part of aging, and the cell will eventually die because it's lost important information that it needs to operate. And this happens in all 46 of the chromosomes and both ends of the chromosome. So she discovered these special ends that she called telomeres. Telo means end, so telomeres. And then, after getting the Nobel Prize for this in 2009, she continued her research and discovered that there's an enzyme, a protein, in certain cells that allow the cell to copy that end, so it doesn't lose the end of its chromosome. This is found in infants, in babies. So as embryos are growing into fetuses or growing into infants, these chromosomes stay the proper length. They don't lose anything important. And it's also found in cancer cells, which makes cancer cells eternal. They don't die like normal cells do, because that end keeps getting replicated and it's not lost like in healthy cells. So that was real interesting information, and her findings showed that if you can decrease the level of telomerase, which is the name of that enzyme, in cancer cells, then that will cause them to die. So it's now one of the new anti-cancer therapies, is to help the body eliminate the cancer cells without radiation and chemotherapy and stuff like that. So that's a good thing. And then for us, something else that's wonderful about telomerase is that if we can introduce this into normal cells, then our normal cells won't lose important stuff either, and we might live longer. Now, some days that's a great idea, and some days that's not a great idea. But it does keep the telomeres from getting too short. And there are high telomerase activity in youthful cells, and it decreases with aging. So if we could do something to promote it so that we had a little bit more of it a little bit longer, we might live a little longer.
I know.
So, thanks to Ms. Blackburn for that. There, Elizabeth Blackburn. She's still alive, and she is still researching. So who knows what else is coming. All right. Now let's talk about genes and how they're related to chromosomes and how they're related to traits or the characteristics that make us up. Now, it's important to know that you can see a lot of traits, a lot of characteristics of people, but there are also a lot of chemical ones going on inside of you that you never see. For example, if you have insulin deficiency and diabetes, You don't see that. These are chemical things going on inside of you. So, traits encompass a whole bunch of things, not just how tall you are, what your hair color is, and those kinds of things. Okay. It is thought that there are about 25,000 genes in human chromosomes, on human chromosomes. The original thought was, well, surely it must be 1 million because we're so complicated that there's got to be a lot of them. But at the most recent count, it's about 25,000, maybe even a little less than that. And some of these are duplicates. Sometimes we have 3 or 4 copies of the same gene. Lots of nuances that I'm not talking about today. Okay, so genes are regions of chromosomes. So there are lots of genes on each chromosome. Okay? Almost sometimes the analogy is to use beads on a necklace, that there's lots of them there. And each one, our definition of a gene is that it's a region on the chromosome where these nucleotides, these A's, C's, G's, and T's, contain the information to produce a trait or characteristic. Now that's real simple because there are several traits and characteristics that require more than one gene to accomplish them, and if that's all it took, we would have things like Alzheimer's and Parkinson's all solved already. So, it's a lot more complicated than that, but the general definition of a gene is the amount of DNA it takes to produce a characteristic. Okay? These can be short, a couple hundred nucleotides, and it can be thousands of nucleotides long. So not all the beads on the necklace are the same size. Okay? If you untwist the double helix, you find the nucleotide sequence there. And what I think I did not point out before is that they match up very specifically, that every time there's a C on one side, there's a G on the other side. And every time there's an A on one side, there's a T on the other side. And going farther up, there's the A's over there, but the T's over here. Always the partners are the same. So a series of those contain the pattern to make a protein, polypeptide, a protein, that then causes the trait or the characteristic. Okay, so why is this interesting? In the 1990s, it was discovered that bacteria naturally make little molecules, little protein molecules that act like scissors, and they will cut DNA at specific locations. And there's lots of these scissors, and so you can cut up a piece of DNA lots of different ways and know where you've cut. But you are at the mercy of the DNA, the scissor. The scissor only recognizes one place to cut. By cutting it, you can potentially remove a damaged piece and put a healthy piece in its place. Okay. But what happens if the piece you want doesn't match the cutting of the scissors that you've got? I mean, you got lots of scissors, but it might cut on one end, but it might not cut on the other end where you need it out of there, kind of thing. What Jennifer Doudna, with her 2020 Nobel Prize, figured out was how to use a molecule, 2 molecules, in a way that she can specify exactly where she wants it to cut, and it will go cut right there. So you can imagine that that's pretty exciting for scientists and health-related people who know that there's a piece of DNA that is defective that they would like to remove. Imagine this happening in an embryo so that the child never has diabetes because the insulin gene is replaced. Where this is happening, the cutting edge of it right now is with sickle cell anemia, and they've been successful in cutting out the sickled mutant gene and replacing it with a boy in New York City. So it comes, these molecules that she found come from a bacteria, Streptococcus bacteria. So when we think strep, we think, oh, sick, you know, I don't want any strep. But she was able to use it in a helpful way as an experimental organism. And the technology is called CRISPR. So if you read or heard about CRISPR, this was hot stuff back in 2012 when she discovered it. And I have her book that I meant to bring, forgot to bring, would be glad to loan it to you. It's called A Crack in Creation. And she's just amazing with the way that they discovered it. So this little protein called Cas9, and then a particular little RNA that's used as a guide for it, and they can tell it exactly what nucleotide sequence they want it to snip, and it will go in and do that. And they can then have snipped a healthy piece out of a different cell and put it in. Oops. And she got a Nobel Prize. Okay. Why is this important? Well, the treatment for the diseases already, for ones that are caused by just one gene mutation or one gene difference, sickle cell anemia, like I mentioned, but also cystic fibrosis, Huntington's disease, and in August last year, the National Institute of Health reported using it for Tay-Sachs. In September of this past year, a similar report was made for replacing the damaged insulin gene with a healthy one for treatment of diabetes. So we are close on these disorders that are caused by just one gene. Now, we're talking not Parkinson's, not Alzheimer's, which are multiple genes involved, but this is the threshold, this is the beginning, when it's one gene, so they can find one damaged piece of DNA and they know which chromosome it's on. They can go in there and snip it out and replace it. So again, an exciting finding by a female scientist. Moving on to the other part of what DNA can do. We've talked about gene editing, which is manipulating the pattern. And now this is what happens after the pattern is used to make the RNA. So protein production happens in 2 steps. Again, this is 4 lectures in my class, so we're doing just this much. Okay. The first part is using the one side of the DNA molecule to be the pattern and making RNA, in this case, messenger RNA. Now, there's at least 6 or 8 different kinds of RNA, but we're just talking about one. Okay. And this happens in the nucleus of the cell, and then it gets processed. It gets parts of it snipped out and other parts hooked together. It gets little chemical groups added to the bases, so it gets modified before it's ready to leave the nucleus. And part of that is for protection, because once it gets out into the outer part of the cell, it's at risk of getting digested because it's an unusual molecule that the cell isn't used to seeing. Once it is successful in getting out of the nucleus, The last step in making the protein is called translation. And here, the mRNA is used as a pattern to put the amino acids together to get the train in with the right cars in the right order and the right length to do whatever job it's supposed to do. So maybe our example is insulin. Okay. Caitlin Carrico, whose Nobel Prize is very recent, 2023, is the scientist who did the lead research for this. She did two things for mRNA that made it really helpful. She started in 2006, and she looked at how the cell normally modifies or changes the nucleotides in the mRNA before it goes out of the cell, or out of the nucleus. So she studied that and was aware that unmodified mRNA causes inflammation when it is put in healthy cells. So if you were to use that as part of a vaccine and inject it in your cells, it would cause inflammation along with whatever production of antibodies that it was hoping to do. Okay. So she developed a method then to modify the mRNA in such a way that it would not cause the inflammation. And then she developed a way for it to be delivered by packaging it in little fat modules, like I'm thinking Star Trek and the pods that would go out from the spaceship to, not Earth, but whatever planet they were going to and come back again. So it's like the mRNA is in this protected protein, or lipid fat layer, and then could go out into the cytoplasm and even out of the cytoplasm to be collected to turn into a vaccine. So this was a technology behind the mRNA vaccines for COVID that biotech and Moderna were able to put together so quickly, because it did not cause the cell to have an immune reaction to the messenger RNA, and it was able to then make the proteins necessary to destroy the COVID vaccine, or the COVID virus. Okay. So in addition to this, since that was all going on in 2020 and 2021, when we thought there would be no cures, and then all of a sudden there was protection available, and it happened very quickly for approval. Since then, mRNA is being used for treatment of some cancers and cardiovascular disease and some of the metabolic diseases. So, another great step forward by a female scientist. Okay. I've talked quickly and a lot. Anybody have any questions? I have two hands, I'm so pleased. Joy.
I have two questions. The cutout part, where does it go? And with the mRNA, we know that some people had bad reactions to the COVID vaccines. Is there any way to avoid that in the future? We didn't hear a question.
All right, the first question, because she's asked me two, what happens to the part that gets cut out and when they're doing gene editing, removing damaged DNA, it just gets destroyed by enzymes that are also in the nucleus, so digested. Then, I don't have an answer for people's reaction to the negative reaction, of which there was a lot to the mRNA vaccines. I don't know the percentages as to how many people were that way, and I don't know if they've done demographics to know if it's a particular population that responded in that way, because if so, then there may be something in their DNA that makes them resistant to this vaccine. Barbara?
Back when you were talking about cutting the scissors, and I think you mentioned that going in when it was an embryo, that's when that can happen. Is that the only time that can happen?
No. And in fact, with the sickle cell that they've just completed successfully, they're doing that in cells in the blood-forming tissue and bone marrow, because it's red blood cells that are affected by sickle cell. And so if you can plant healthy producing red blood cells, bone marrow, in the bone marrow, stem cells, that will then make healthy red blood cells, the embryo would be a dream. I mean, that'd be way down the road. But in the same way with insulin, that those are being planted in the pancreas and the Isles of Langerhans that make the insulin. So if you could put healthy cells in there or fix those cells. But no, not at all. It's happening in adults, for sure. More?
Yes, I have a second question. I just read an article like 2 days ago that I felt so educated after I read it. And back in the beginning, in one of your slides, when you talked about the characteristics, and this article was about how you may be have the genes or hormones and all of that to be a female, but you could also have the hormones within you that are not visible that would be male, or vice versa. And it gave me a new understanding of human nature.
That's a whole other topic that we didn't get to today. But, and part of it, Joy asked me about the X and Y chromosomes early on, and we didn't talk about that at all either, but the gene, the single gene that determines maleness is on the Y chromosome. There's not very many genes on that Y chromosome. It's like 70 maybe compared to the many that are on the others. But that one is there, and there's another gene on the X chromosome that interacts with it to determine maleness. And again, things can misalign or fall apart there and give us the unusual situations that we see. Coming back, Barbara?
What kind of research have you personally been involved in?
Nothing human. Nothing human. Yeah. I think, I mentioned when I was doing my journey in life that I was on the early edges of this, the early 1970s and middle 1970s, and what we were doing was developing tools that could then be used by these scientists later on. And what I was doing was digesting the parts of plants, doing plant tissue culture, which is now really common in Apopka and every place else where they're building or growing organisms where they want all of them to be exactly the same. But I was working on a tool is what I did. Peter.
See, this may be a little simplistic, but Mary and I and Joe Burns and Linda are all raising butterflies from caterpillars, monarch caterpillars in our apartment, in a little tent,
Right.
and watching it go through all the stages.
Mm-hmm.
Is it fair to say that all the genes and chromosomes and whatever are in the caterpillar to begin with that turns into a butterfly?
Yes. And they were in the egg before that ever hatched into the caterpillar. Yeah. And I guess the way to approach that is to say that in all these 25,000 genes, they're not all working at the same time, that they turn on and off with regulator molecules that are in the cells. And that's why you can have 2 stages of life that are like totally different from each other. The caterpillar, well, the egg, then the caterpillar, then the chrysalis, and then the adult butterfly, because of which genes are turned on at what point. Oh, and there's some great stories about mutations that cause organisms to stop in one form and never turn into the adult, for example. Oh, you can do mean things with radiation. Did that answer it? Joy?
Well, we recently had a discussion on how we figured out what the purpose of the art was. Now we're talking about DNA. What is there left to examine?
There is so much about DNA that we still don't know, like what turns them on and off, these genes, and the multi-factor disorders like Alzheimer's and Parkinson's. There are genes that are interacting with each other, to or on when they shouldn't be or off when they should be, kind of thing. There are a lot of puzzles along that line, and of course we're most interested in our health, so we want to know about that. But then there are all kinds of genetic things related to crops and food supply and those kinds of things. Even creating, who did this recently? Some little organism, I think maybe a bacteria that eats plastics, that's eating up, plastics. And again, can you grow more of those? Can you affect a bacteria that can't do it in such a way that it'll start to do it, or its offspring will do it? So there's still lots of that. Chuck?
Is telomere length related to longevity?
Yes.
And if so, how can we increase the telomere length?
That's it, exactly. That's it, exactly. Has to do with telomerase, that enzyme. If you can cause the cell to turn the telomerase making gene on, even in older cells, that will take care of that. Yeah. And that's, as you can imagine, a hot area of research. Yeah. Joy? Just checking to see if Barbara, did you have your hand up?
Yeah.
Let me get Barbara and then we'll come back.
Halfway, it's wonderful that they're doing this gene splicing.
Barbara, I can't hear you, just a minute.
It's wonderful that they're doing this gene cutting to help sickle cell and other diseases. And what do you think they are going to use for Alzheimer's, Parkinson's, and other diseases?
I'm thinking still a far distance away because they're so far limited to just disorders that are caused by one gene, and Parkinson's and Alzheimer's are many genes involved, and another one, schizophrenia, that's another one that is multiple genes, and when they figure out how to untangle that, then maybe they can figure out what to snip out. But that's why we keep raising money, right? So that people will keep researching it. Barbara?
What are the areas that are most likely right now, to have solutions? What's hot? Prediction?
I don't know if there's an answer to that because things come up in the most unlikely places. We would have to go around to the places where the major researches are happening and say, so what are you looking at? And sometimes it's totally serendipity. You transfer to a new school, and you meet a new colleague who's studying something totally different than what you are, and all of a sudden it comes together and go, oh, I can use that. And that's how so many of these discoveries occurred, just because people talk to each other. Communication is crucial. So Joy's got one more, and then I'm going to go to the final slide because Stan's here, and I've got 6 minutes.
Okay, you were talking about the impact on health. Is there anything that impacts characteristics like being a genius or being a criminal, etc.?
No, and there's another multifaceted thing, and I didn't even address the fact that your genes give you a blueprint, but the environment that you live in, nature versus nurture, there's at least an equal amount of effect from that to the genes. Genes kind of give you a framework, and then it's like you have blueprints on the house. Well, maybe you follow it to the letter, or maybe you make a little change, or that kind of thing. And so it's that thing has a lot of nurture in it, as well as the genetics. Well, thank you for listening. I have one goodbye slide. Do you recognize her? This is the first female scientist I ever saw. Watching Star Trek in the 1960s. And that let me know, oh, girls can do this, right? And that's her quote. Science is not a boys' game, it's not a girls' game, it's everyone's game, everyone's game. So thank you for coming.